Hi and a very good morning to Doctor. Today our group will be discussing on optical biosensor. But first I will be introducing our group members, which is Nur Shamimi, me myself is Zina Shoha, Aida Sarah Samhana, and also Muhammad Hafiz. So our presentation will be discussing on the introduction of optical biosensor, the mechanism mode of optical biosensor, advantages and disadvantages of optical biosensor, application of optical biosensor in terms of environmental monitoring and also medical diagnosis.
Good morning and hi doctor. My name is Aida Sarah Samhana binti Muhammad Sufian. Today, I would like to present with you about optical biosensor. Alright doctor, why our group choose optical biosensor? Because it have many advantages and application doctor. My group members will explain about mechanism, application of newly developed biosensor and others. Let me start first, doctor. What is optical biosensor? Let me think first. I think optical biosensor is one of such nanobiomolecular devices that has a potential to pave a new dimension of research and devices fabrication in the field of optical and biomedical field. And beside development of optical biomolecular device is a new more towards revolution of nanobioelectronic. Beside that, doctor, for second principle, which is principle label-free optical biosensing, with this one is without any reporter, the immobilization of biorecognition element is the same as the label-based technique. Same like I explained previously, doctor. Then, any biomolecule attachment to the surface of that sensor modify the index of refraction in the immediate vicinity of optical device. Then, the target molecule bind to the surface of the functionalized optical devices, forming a thin layer and further modifying the refractive index. This change can be monitored by tracking the change in the natural frequency of vibration or the resonant frequency. Alright, that's all for principle of optical biosensor, doctor. Furthermore, at the bottom of slide have the reference for my slide, doctor. Thank you.
Mechanism or mode of optical biosensing. Optical biosensing can be broadly divided into two general modes, label-free and label-based. Example of label-free is surface plasmon resonance (SPR), where it do not require the use of label to facilitate measurement. Example of label-based, it requires label such as enzyme and fluorescent or radioactive molecule attached to the target analyte. As we know, the basic objective of an optical biosensor is to produce a signal which is proportionate to the concentration of measured substance or analyte. And this is the basic principle of a commonly used label-free optical sensing technique, surface plasmon resonance (SPR). SPR instrument use a detection scheme called a crash point configuration. In this setup, a light source passes through prism reflect off at the backside of the sensor chip surface and into the detector at a certain incident angle known as resonant angle. Light is absorbed by the electron in the metal film of the sensor chip, causing them to resonate. These resonating electrons are also known as surface plasmon, which is sensitive to the surrounding environment. The result is an intensity loss in the reflected beam, which appears as a dark band and can be seen as a dip in SPR reflection intensity curve. The shape and the location of the SPR dip can be used to convey information about the sensor surface. SPR technology is commonly utilized for the study of molecule-binding interaction between the free analyte molecule and solution and probe molecule, which are linked to or immobilized on the sensor surface as the molecule binding event. They take place the angular position of this dark band shift, and the shift in the reflectivity curve can be observed. D is a direct method of detection which avoids the drawback of label. Another powerful aspect of SPR technology is in its ability to observe the time-dependent binding interaction between the molecule by monitoring the change in SPR response over time. And it can also study the kinetic of molecular binding event. In first example, an analyte with low affinity to the immobilized ligand on the sensor surface is introduced, and therefore negligible binding interaction is observed. However, when analyte with high affinity to mobilized ligand is introduced into the system, then binding events are more readily observed. Initially, there are many binding sites available, resulting in rapid increase in SVR response occur as an analyte begin to bind to the ligand. While sample is continually delivered to the sensor surface, analyte molecule continue to bind in the number of available binding sites, decrease corresponding to the decrease in binding rate. As the SPR response level of the system approach equilibrium, the number and the molecule binding and unbind become equal. When no analyte is introduced into the system, the molecule will continue to unbind, resulting in decrease in SPR response. The association constant Ka can be extracted from the behavior of the binding response, and likewise the dissociation constant Kd can be extracted from the unbinding response. The ratio of these two constants can yield the binding affinity of the system. The information that can be obtained from the SPR serves as simple, precise, and label-free technique for quantitative analysis of molecular binding interaction.
Hello doctor, my name is Muhammad Hafiz Muhammad Said.